Module 16, the reproductive systems, fertilization, development, and parturition. I already mentioned that at fertilization, an individual's life begins. Another word for this is conception. You can see in this image that many sperm cells are attempting to fertilize the much larger ovum. The first one to make it through the outer layer will fertilize the egg cell. At this point, the fertilized ovum is called a zygote. The word zygote means paired, and it refers to the pairing up of the two sets of 23 chromosomes from each gamete. The zygote has 23 pairs of homologous chromosomes, or 46 total chromosomes. The sperm cell that fertilized the ovum really only contributes its DNA information. It doesn't have much cytoplasm to bring to the zygote, nor does it have any major organelles. So most of the cytoplasm in the zygote comes from the ovum, as well as all the mitochondria with their mitochondrial DNA. That's why we can say that all mitochondrial DNA is inherited from the mother. In the last section, I mentioned how the ovum is fertilized in the uterine tube. Therefore, the zygote has to continue traveling down it into the uterus. It moves along due to the peristaltic movement of the smooth muscle of the uterine tube as well as the cilia there. Even though this cell is a single cell, the zygote has all the genetic information it needs to completely develop into a mature human. Its gender is determined too. You see, because the sex chromosomes of a female are both X's, all ova will have an X chromosome to contribute to the baby's gender. But because a male has an X and a Y sex chromosome, half of the sperm will have an X chromosome to donate, and half will have a Y chromosome. If an X chromosome is in the fertilizing sperm cell, then the zygote will have two X's, and the baby will be a girl. If a Y chromosome is in the fertilizing sperm cell, then the zygote will have one X and one Y, and the baby will be a boy. This is the point when fraternal twins can happen. In most cases, a single ovum becomes fertilized, but occasionally a woman will ovulate more than one ovum. If both become fertilized, then two babies will develop. They won't be genetically identical, though, because two different ova were fertilized. So, though they will develop and be born at the same time, they will not have identical genes and will appear like any other brother and sister. Identical twins are formed in a different way. After an ovum becomes fertilized, the zygote begins to divide or cleave via mitosis. In fact, within about 18 to 36 hours after fertilization, a zygote begins to divide into two cells. This begins the cleavage stage. These two cells then each divide, making 4 cells, and so on, and so on. Now, the embryo doesn't grow in size during cleavage. It is just making more and more cells within the same sized sphere. In rare cases, in the early stages of cleavage, the cells that are dividing don't stay together well. If they separate, they will each continue to divide, forming 2 separate embryos. That is how identical twins form. Both individuals came from the same zygote, so they have identical genetic information. But they are not exactly alike. That's because genetics don't determine everything about a person. Various developmental, environmental, yes, even within the same womb, and even spiritual factors contribute to a person. Well, after a number of cells have been produced in the cleavage stage, a morula is formed. This happens about 3 days after conception, and the morula is still in the uterine tubes. The next developmental stage is the blastula stage, which occurs only 4 to 7 days after fertilization. The morula cells continue to divide more and more, and now the embryo grows in size. The cells push outward and form a cavity inside that is filled with fluid. This cavity is called a blastocele, and this results in a blastocyst. It has one layer of outer cells called the trophoblast cells. That is the feeding layer, which will eventually develop into the placenta, umbilical cord, and amniotic sac surrounding the embryo. The feeding layer will give oxygen and nutrients to the embryo through the placenta that it forms. Now, there's a small flat group of cells on one side of the blastocyst, which is called the inner cell mass. That is the actual embryo. The embryo remains in the blastula stage to about 7 to 9 days after fertilization. Then the blastocyst implants into the vascular layer of the endometrium of the uterus. This is called implantation. Implantation is the burrowing of the blastocyst into the endometrium. This usually happens high up in the uterus. In order to implant, the trophoblast cells have to secrete digestive enzymes so the blastocyst can burrow into the tissue. At this point, the embryo can receive nutrients and oxygen from the mother's blood. Once the embryo has implanted, another cavity forms from the trophoblast cells. It's called the amniotic sac. Now the embryo is in the gastrula stage. The inner cell mass, which is the actual embryo, starts to change its shape. Up to this point, all the cells of the embryo are identical, but now they begin to differentiate, forming 3 germ layers: the ectoderm, mesoderm, and endoderm. These 3 germ layers will form all the tissues and systems of the body. The ectoderm forms the skin, nervous system, and some of the glands. The mesoderm in the middle eventually forms the circulatory system, muscles, and most bones. The endoderm eventually forms the lining of the digestive tract, the respiratory system lining, most of the glands, and the digestive accessory organs like the pancreas. After the gastrula stage, the next stage is the nerula stage. This is when the neural tube forms. What happens is that the flattened 3 layers of the embryo begin to fold inward, and both folds come together on top to form the neural tube. This is where the brain and spinal cord will form, and it happens only about 3 to 6 weeks after fertilization. The next stage is the organogenesis stage, and I'm guessing you can figure out what happens in this stage. Organogenesis is the formation of organs in a developing embryo, and this stage occurs from the 2nd to the 8th week after fertilization. This is when all the organs of the body are formed, though many may not be completely functioning. At the end of this stage, the baby is no longer called an embryo, but is called a fetus. You can see that by the end of this stage, development has occurred rapidly. After just 9 weeks, you can identify fingers and even specific bones, including the forearm bones and the jaw. The fetus is surrounded and protected by an amniotic sac. So how does he or she get the food and oxygen for growth and survival? Well, most mammals produce a placenta. This is an organ that lets nutrients and wastes to be exchanged between the mother and child. And this system doesn't mix the 2 individuals' blood. Rather, the mother's capillaries run alongside the baby's capillaries in the placenta. Thus, nutrients and oxygen can pass out of the mother's capillaries and into the baby's. Wastes can then pass out of the baby's capillaries and into the mother's for removal. The capillaries in the placenta travel through the umbilical cord so the fetus can receive the nutrients and oxygen it needs. The placenta also has another function. It makes a lot of hormones, including the very important estrogen and progesterone. In fact, it's the placenta that is the primary source of these 2 hormones during pregnancy. Human gestation, or a full-term pregnancy, takes an average of 8 months and 3 weeks from conception to birth. Then it's time for parturition. Parturition is the process of childbirth. This process begins with labor, and it begins by a release of hormones from the fetus. Stress hormones are produced by the adrenal cortex of the unborn child because at the end of gestation, there's a greater and greater need for more nutrients and space to grow. The stress hormones move through the blood into the placenta and into the mother. This causes a lowering in her progesterone levels. Then estrogen levels increase, as well as oxytocin, which is a hormone that comes from the posterior pituitary gland that causes the uterus to contract. The contraction of the uterus causes a positive feedback system, which further increases uterine contractions. These muscular contractions cause the fetus to push against the cervix, which causes this muscle to stretch. The cervix has receptors that continue to send messages to the spinal cord, which sends more messages to the hypothalamus. As long as the cervix has not stretched enough to allow the baby to pass through, the hypothalamus will continue to secrete more and more oxytocin. Once the cervix stops stretching, which is when the baby is born, then the level of oxytocin will begin to decrease. Labor can last anywhere from an hour to 20 or more hours. It varies from woman to woman and pregnancy to pregnancy. But for the most part, it always lives up to its name. It's a laborious feat, producing a wondrous result. Module 16, the reproductive systems. Final project, fetal pig dissection. Dissecting a fetal pig is a great opportunity for you to get a good look at all the organ systems of the body. Now that we've covered or worked our way through all of those systems, so we're going to begin by looking at the external features of the pig, particularly in its mouth area. This is a mammal, so mammalian structures will be similar. You can see the hooves. This is the mouth area. This is the tongue. You can actually open the mouth a little bit, see the hard palate, which is the ridgy section. Let me see if I can use my scissors to help pry that open a little bit more. You can actually see tiny little teeth that are forming there. And that ridged hard palate, the nares. the ear flaps right here. And we're going to begin our dissection by looking at the internal cavities, the thoracic cavity and then the intestinal area. And I want to use my scissors for the most part, and I'm going to do this. I'm going to cut, let me just show you, a shape across this area, and then down, and then I'm going to cut another T at this point, right at the arms, and then down, and then a final T, so we can see all the different sections. So I'm going to begin right here in the middle. Let me cut across. And remember, as we talked about before, when you're cutting, it's always good to lift up a little bit with your scissors so you're not damaging the internal structures. Then I'm going to cut straight up. And then another T over here this way. So you can see the thoracic cavity first. And you can go slowly. You don't have to rush through this. You can see muscular tissue, so I'm going to cut through that because we want to get into the cavity. Just be patient and go slowly with this. Let me cut a little bit higher here so I can see what's going on up here. All right. Better readjust my gloves so I can see what I'm doing. Okay. Move this muscular material here over. All right, we're getting there. Now let me see if I can utilize some pins. Okay, so now what we're looking at is the upper part of the body cavity. And you can notice here the part of the lungs. The lungs are the softer tissue on each side that I had to cut through to get to this spot. You can also see the ridged trachea that goes to the lungs. And then below that will be the esophagus. Let me see if I can get in below there. Kind of tweezing the tissue. And if you can't find it that way, maybe what we'll do is we'll look for the stomach and work our way back. Right here, this soft material right here separating the two is diaphragm right here. Okay, now let me go ahead and open up more of the cavity. Turn it this way. And then once I do that, I'm going to pin those flaps open so we can see better what's going on. Okay, before I open up this side of the flap, I want you to notice this is the umbilical cord, and can you see how it extends into the abdominal cavity, and that's where all of the nutrients and wastes for exchange are. So I'm going to cut around that so you can see it a little bit better, but I want you to notice that before I move this flap over. There we go. Okay. Let me reorient ourselves here. Okay, so now we can see much more of what's going on. This large, tough material right here, this right here is the heart. Let me use my non-pointy probe, my blunt probe right here, and you can see it's much more tough.
So right here, these flaps right here, you can see are the lobes of the lungs on each side.
And now we're going to look a little bit lower. Okay, and I was incorrect earlier. This right here is our diaphragm right here. Okay, this material right here separates the heart cavity from the lower part. So this is the diaphragm. Okay, and so then we're going to continue down. This area right here below this liver material, this is the stomach. Nice and soft. And if you'll follow the stomach upward, you'll go to where the esophagus is. So I'm going to see if I can't get behind the liver here. And there you can see my probe underneath it. There's the esophagus going up, on its way up to where the mouth area is, behind the heart and lungs and up into there. So the lungs were up here, just reorienting you. There's the trachea, what's left of the trachea, and then our heart. We have our liver, and we have our stomach. And then following the stomach, we have our small intestine, which is right here. This is the small intestine. Okay, and then you can, if you want to find the end of the stomach where it moves into the small intestine, you'll find the duodenum, which is a straighter area. That's the duodenum right there, or duodenum, depending on your tomato pronunciation of the duodenum. Okay, and then we have our small intestine, which of course takes up a large portion of that abdominal cavity. And then the small intestine then moves to the large intestine, which is a little bit different in shape. And I'll see if I can find it. Here it is, right here. The large intestine is kind of spirally oriented together. And so that gives it much more surface area in a small space. So this is the large intestine right here. The small intestine, I also want to point out, is connected together by what's called mesentery tissue. And that's what you can see right here. This tissue, let's move that flap over. This tissue right here holding all the small intestine together is mesentery. And it provides, it's full of lots of blood vessels for nutrient exchange. Because remember, the intestine is where food is absorbed, nutrients are absorbed, and it goes into the bloodstream. And so this mesentery holds the small intestine together, as well as has lots of vessels for that exchange. All right, and then let's see if we've got anything else here worth looking at. Okay, I wanted to talk about the sex of this specimen. This is a female, and so what we can see, if you can look right here, is a Y-shaped organ right here. This is the uterus, and so if we look on both sides over here, let's see if we can find the ovaries. This is a small specimen, so I may not be able to see those in this specimen. But if you were to follow that Y up, there we go, there's one ovary right there. Perfectly formed. It's such a tiny specimen. Okay, and then we also, right here, this almost looks liver-like in its shape. This is the spleen. Remember, the spleen plays an important role in our immune system. And so this is the spleen coming over the stomach right here. All right, we've covered some of the basic portions of the organs of the body. what's really interesting about this is as you have a better understanding of all those organ systems, you can take your time and slowly go back through and try to make sure you're oriented on all of these systems and how they're connected. And you'll also get a better understanding of some of their interconnectedness as well. So we went over a really quick look at the fetal pig and the organ systems of the fetal pig, and notice that it's very similar in design to other mammals, including humans. So by doing this dissection, you get a really good look at all of the organ systems of the body. This is the final project, so take extra time here. Make illustrations of each system if you can, using the guide that's in your text, as well as some of the illustrations of the organ systems that you have in your text as well. Identifying all the parts that you can find. Maybe if there's something that you're unsure of, make this kind of like a sleuth. You're some sort of detective trying to find out, and you can deduce whether certain organs are going to look like one thing or another based on the diagrams as well as what you're seeing here. Really enjoy this exploration of the systems of the body with the full understanding that our extremely complex body is perfectly made and perfectly designed. Well, this completes our journey of learning about the human body. In covering all 11 of the organ systems, you've discovered their precise arrangement, design, and critical roles in keeping you alive and healthy. They are perfectly created to grow, maintain themselves, respond to the surrounding environment, and react to changes. And our bodies do these jobs day in and day out for the most part without our having to be conscious of them. Because there's much, much more to the human body than its physical design, right? Humans are able to live and work to perform life-critical activities such as breathing, but they're also able to sing, dance, and understand things we call beautiful. Our God doesn't just create food to keep us alive. He gives us food with awesome flavors so we can savor and enjoy the process of eating. He gives us other senses to admire beautiful artwork. He makes us able to communicate and build relationships with each other and with Him. Doesn't this give you a greater understanding of the complexity of God's creation? Our bodies are so much more than machines. We can truly say that no matter what awesome designs that humans are able to create, they in no way will rival that of God's perfect creation.